Hello, and thank you for your attention to Humans and When Neurological Surgery, Chapter 377, Genetics of Traumatic Brain Injury. My name is John Yue from the University of California, San Francisco. Genetic determinants of patient response and outcome in traumatic brain injury are multifactorial. Genes involved in neuronal repair, neuroinflammation, cell survival and apoptosis, neuroprotection, and neurotransmission are known to influence TBI outcome. The diverse mechanisms by which these genes exert their effects underscore the challenges in understanding TBI and also suggest the existence of many unexplored treatment modalities. Recent advances in data standardization have facilitated data pooling across multi-center clinical trials, enabling the identification and validation of high-priority candidate genes for TBI recovery. Heritability, or genetic susceptibility factors, is intimately associated with disease progression in a number of neurological disorders. Genetic susceptibilities are also emerging in TBI with relationships, especially to injury severity and recovery. Single nucleotide polymorphisms, or SNPs, are nucleotide substitutions at a single base pair. They're a common source of inherited genetic differences. SNPs may arise within both coding and non-coding sequences and may alter protein function and variances in their level of expression, respectively. The improved understanding of how genetic variants influence prognosis and outcome in TBI and validating new candidate genes will allow clinicians to better identify and further stratify patients for tailored therapeutics. A few key points. SNPs may be implicated in neurodegenerative, inflammatory, neurotransmission, apoptotic, and neuroprotective pathways. They may also have profound effects on overall TBI recovery and also in select subpopulations of TBI. For neurodegenerative pathways, we will focus on apolipoprotein E, epsilon 4, and how this specific allele increases the risk for beta amyloid aggregation and secondary injury across a number of populations. For oncogene pathways, we will focus on B-cell lymphoma 2, or the BCL2 allele, which induces dysfunctional apoptotic regulation that causes accelerated cell death. Importantly, the G allele, which is associated with a number of poor outcomes, will be discussed. For dopaminergic pathways, there are several alleles to discuss. The ANK1 RS1-800-497 variant confers decreased dopamine receptor D2 expression in the striatum, which may lead to a number of impairments. The catechol-methyltransferase, or COMT variant, confers less enzymatic activity to the COMT protein, which may increase dopamine levels at the synaptic cleft. For serotonergic pathways, the serotonin transporter 5-HTT short allele confers less transcriptional activity to the 5-HTT, or the serotonin transporter, compared to its long alleles. The short allele may increase the risk for major depression in certain psychiatric evaluations. However, it may also confer less risk for depression in moderate to severe TBI at 6 months. Therefore, this indicates a TBI-specific pathogenesis of depression, as well as gene and injury interactions. And finally, data standardization, the pooling of large prospective data sets, can help achieve the statistical power for exploratory and validation studies that can determine high-priority candidate genes for TBI recovery. We will begin by discussing neurodegenerative pathways. Here you see a figure regarding the different types of pathways that genetics and genetic polymorphisms are implicated in. For this portion of the talk, we will talk about apolipoprotein E and its interactions with beta amyloid deposition, as well as BDNF, or brain-derived neurotrophic factor, and its downstream effects. Apolipoprotein E is a protein in lipid metabolism and a ligand for multiple receptors such as the LDL receptor. It is required for beta amyloid deposition. The ApoE genotype is a strong predictor of risk in Alzheimer's disease. There are 3 common isoforms, epsilon 2, epsilon 3, and epsilon 4, that differ by the presence of a cysteine or an arginine at 2 sites in the protein structure. Importantly, the epsilon 4 isoform has enhanced aggregation with beta amyloid, as well as mitochondrial toxicity, increased tau phosphorylation, and vascular injury. Epsilon 4 homozygous individuals have a 33-fold increased risk of Alzheimer's disease. Regarding TBI outcome, Teasdale et al. Showed that nearly 60% of epsilon 4 carriers had a GOSE of 1 to 4, which was death or severe disability at 6 months post-injury. Numerous other studies have shown that the epsilon 4's association doesn't end with just poor outcome, but also increased hospital length of stay, as well as slowed recovery over as long as 2 years after injury. In military studies, epsilon 4 carriers have associated with decreased memory and processing speed, as well as increased mood disorders after traumatic brain injury. And it is thought that ApoE may exert effects in the frontal subcortical regions to accelerate early neurodegeneration. In civilians, epsilon 4 carriers have been associated with long-term memory deficits at 6 months after traumatic brain injury, as you can see in this figure below. In this figure, the red bars signify epsilon 4 carriers, and the blue bars signify epsilon 4 negative individuals. And as you can see, the non-epsilon 4 carriers scored higher in almost all scales of verbal memory performance at 6 months. Furthermore, in the sports literature, verbal memory deficits were found to be 4 to 8 times more likely in epsilon 4 carriers with head impacts compared to those without. And here on the bottom right, you see a figure regarding odds ratios for the association of ApoE genotype with poor outcome in different age groups. Specifically, these are for epsilon 4 carriers across different age groups. And as you can see, in younger patients or younger people, being an epsilon 4 carrier actually confers a greater odds ratio for poor outcome at 6 months. Next, we will talk about brain-derived neurotrophic factor. Pro-BDNF is proteolytically cleaved into its mature and more active form, BDNF. BDNF is then released from presynaptic terminals and is implicated in neuronal synaptic growth and maintenance, axonal guidance, dendritic growth, and long-term potentiation. The BDNF RS6265 SNP, where valine is substituted with methionine at the 66 spot of the promoter region, reduces BDNF release in neurons. Carriers of this SNP have deficient episodic memory and impaired plasticity in a number of large trials. Studies in TBI, however, are mixed. McAllister et al. Showed that the RS6265 SNP is associated with slower processing speed 6 months after mild to moderate TBI, as evidenced by the figure here on the upper right. As you can see, methionine homozygous individuals have significantly longer reaction times compared to methionine carriers and valine individuals. However, Kruger et al. Showed that the RS6265 SNP is associated with better executive function in combat veterans with frontal lobe injury. Again, here on the right bottom figure, you see that the black bars, which are methionine carriers, scored higher in almost every category of executive function compared to valine homozygous. Therefore, the thought is that there may be differences in BDNF regulatory effects depending on the region of injury, as well as the temporal time frame after an injury that has happened. Next, we will move on to oncogene pathways. Proto-oncogenes and tumor suppressor genes encode key regulators of cell proliferation. They also include regulators of cell survival and death. Imbalance between cell survival and death pathways may contribute to the evolution of injury and modulate recovery after TBI. B-cell lymphoma 2, or BCL2, is a proto-oncogene that enhances cell survival by preventing permeabilization of the outer mitochondrial membrane. Thus, it prevents the release of pro-apoptotic molecules. TBI survivors have higher CSF levels of BCL2 compared to those who do not, suggesting a protective effect. The SNP RS17759659 variant allele, or the G allele, is associated with poor Glasgow outcome scale in multiple studies. In a multi-center study, GG patients, or G homozygous patients, had a mean of 3 millimeters of mercury higher in intracranial pressure, 2.5 the odds of cerebral edema, and 3.7 times the odds of requiring surgery for decompression. G allele carriers had more frequent ICP spikes as well. And thus, it is thought that increased cellular apoptosis due to dysfunctional apoptotic regulation and decreased cerebral compensation may explain these findings. Next, we will talk about neurotransmitter pathways. The dopamine receptor D2 is a key component in the circuitry underlying executive function and working memory. The DRD2 TAC1A RS1800-497, also known as ANCA1, lies within the coding region of a promoter sequence for DRD2. Patients with the variant, or the T allele, have 40% less DRD2 receptor expression. Using 2 large multi-center TBI studies, the ANCA1 T allele had a dose-dependent relationship with verbal memory and nonverbal processing speed at 6 months post-injury. Furthermore, the DRD2C957T RS6277 variant allele, also a T allele, confers reduced affinity for striatal D2 receptors. Therefore, the variant allele allows for increased capabilities of learning, verbal memory, and cognition. The C957T allele carriers have better verbal memory and delayed recall after controlling for a number of demographic factors than the original alleles. And thus, pharmacologic enhancement of dopaminergic signaling may have beneficial effects for TBI patients. As such, medications such as bromocriptine, which is a D2 agonist, have been shown to selectively improve cognitive ability. Catechol-O methyltransferase, or COMT, catalyzes the methylation of dopamine and other catecholamine transmitters to make them inactive. The RS4680 valine 158 methionine SNP have 4 times less the enzymatic activity compared to the major allele, and therefore, methionine carriers may have actually much higher dopamine levels than valine-valine homozygotes. This SNP is particularly relevant in the frontal cortex, where dopamine transporters are low, because COMT here likely has a more prominent role in regulating synaptic dopamine levels. The methionine allele has been associated with improved cognition, including verbal memory and nonverbal processing speed after mild to moderate traumatic brain injury. Next, the serotonin transporter 5-HTT, encoded by the gene SLC6A4, is involved in the reuptake of secreted serotonin to terminate synaptic transmission. It is the principal target of SSRIs in the treatment of depression. SLC6A4 is modulated by a 44 base pair insertion deletion region, the serotonin transporter gene, linked polymorphism region, also known as 5-HTT-LPR. This region gives rise to either a short allele, the S allele with less functional activity, or a long allele, the L allele with greater functional activity. 5-HTT expression is further influenced by a SNP with adenine to guanine substitution in this polymorphic region that decreases the expression of the L allele. Therefore, the LG allele is expressed at a much lower level than the LA allele. Studies in TBI are mixed. Effects of the S allele have been seen in moderate to severe TBI patients where S carriers were less likely to be depressed at 6 months. The LG allele carriers were less likely to be depressed at 12 months. This shows that the S or the LG allele may confer protection from depression at different outcome time points. In conclusion, variability in target candidate genes has been shown to correlate with TBI outcome, implying that a personalized, genetics-based approach to TBI prognosis and treatment is possible. Advances in data standardization have enabled large multi-center clinical trials to achieve adequately powered discovery and validation samples in order to elucidate high-priority genetic polymorphisms in TBI across neurodegenerative, inflammatory, apoptotic, dopaminergic, and serotonergic pathways. A deeper mechanistic understanding of how genetic polymorphisms may alter patient responses to TBI is necessary to develop appropriate rehabilitative and pharmacologic treatment strategies. Technological advances in neuroimaging, such as diffusion tensor imaging or functional MRI, may more precisely define the anatomical location and resultant cellular damage from TBI. Such advances may enable further insight into how genetic polymorphisms affect brain function after injury based on their distribution and known cellular activity. The application of genome-wide association study, which is yet to be completed in TBI, will verify how genetic polymorphisms hypothesize to modulate TBI outcome. It can also identify novel polymorphisms that may not be obvious candidates. Finally, the goal of incorporating emerging technologies is to propel the field forward and fulfill the promise of effective, personalized TBI